Today we will discuss about DNA is the genetic material in molecular genetics unit 2 chapter 5. So DNA is the genetic material. In previous classes we have seen the gene as the functional unit of inheritance, and in search of genetic material that was given by Avery et al. An experiment which Griffith experiment bacterial transformation has proved that DNA is the genetic material. After that also there are some confusions because whether DNA or protein is the genetic material, because protein has a complex structure. So protein structure is a 3D structure and it coding for many amino acids. So why the protein can be the genetic material? Why the DNA? So to overcome this confusion, the other experiment was given by Hershey and Chase. So Hershey and Chase experiment. Okay. After this experiment, they had come to a conclusion that DNA is the only genetic material which transfers the genetic material from one generation to another generation. So Hershey and Chase experiment on T2 bacteriophage, that is bacteriophages means the virus which attacks bacteria is called bacteriophages, and it is an important 5-mark question. Alfred Hershey and Martha Chase in 1952 conducted an experiment on bacteriophage that infects bacteria, that is the virus that infects bacteria. Okay, what bacteria it will infect means E. coli, that is Escherichia coli bacteria, which simply named it as E. coli bacteria. When the phages are added to the bacteria, when the phages are added to the bacteria, the phages attach to the bacterial cell wall. That process is called adsorption. And the material, that is the genetic material, was released in the bacteria. After maturation of the phage materials, the bacterial cell got lysed or the bacterial cell destroyed. Okay, this is the normal procedure you have studied in the that is lytic cycle of virus in the viruses lesson in the last year. Okay, and the Hershey and Chase want to observe whether it was DNA or protein that entered the bacteria. So they must know whether the DNA is causing harmful to the bacteria or the protein. For this, they conducted a cross. Okay. So this is a bacteriophage, okay. And you can see the red color. The red color means radioactive labeled structure, that is S sulfur bacteria, that is S labeled radioactive sulfur labeled, that is sulfur 32. So the red color, and the DNA is not treated with the radioactive isotope, okay. So what they are think, that is think this as a bacteria, okay. So they are attached to the bacterial cell wall. Okay, the protein was there, that is they are taken in a culture medium. Okay, culture medium. And the red labeled structure, only the that is DNA was here, and the red labeled that is red labeled means sulfur labeled material was there. Now they are putting the culture along with the culture medium. They are putting it in a blender. Blender is nothing but a mixie. So a mixer grinder, okay, mixer grinder or centrifugation for agitation, slight agitation. What our moms do for chutney making? They put it in the mixie and they grind it, and they will take the supernatant out, and they will keep the sedimentation for the preparation of chutney, okay. Like that, he made it in a blender experiment, that is blender means mixie, mixer grinder. So there are centrifugation. So for light agitation, light shaking. While shaking, what happened means the DNA material was in the sedimentation part, and the supernatant was removed. The supernatant is protein. Okay. Now what happened? This DNA, that is this sedimented DNA particle was enter into the bacteria. And the radioactive material, that is a S-labeled protein coat, is eliminated. What happened? The bacteria was not infected here by labeling, that is only the protein coat was labeled. Okay. So the protein coat was removed and the genetic material only entering into the bacteria. So due to this, due to this, okay, the proteins are not acting as the genetic material. So this one experiment. Same time, they took phosphorus bacteria, that is phosphorus labeled one in the genetic material. You can see this red color inside the bacteriophage. Same thing, adsorption takes place, that is in the culture medium they put it. And blending means a slight, that is like in a mixie they are giving putting agitation, shaking, no, okay, like that a slight blending, that is slight shaking takes place. The genetic material was in the plate, and now this genetic material was allowed to infect in the bacterium. Okay, bacterium. And the another things, that is only the sedimented part, that genetic material DNA was taken and the protein coat was eliminated as a supernatant. Okay, so what happened now? So this genetic material allows, that is the phage genetic material allow the bacteria to infect. While infecting, the bacteria cell and the bacterial genetic material destroy, and the phage material inside the bacteria was synthesized and mature and releases outside. So this when the, so here the DNA is the genetic material. When they labeled the protein coat, that doesn't cause any harm to the bacteria. Okay, any harm to the bacteria. But here the DNA was labeled with phosphorus and the infection was occurred here. So this proves the DNA is the only genetic material. And we can say one more, that is in other way also we can say. So what happened means the labeled protein coat and the labeled DNA mixed and allow the bacteria to infect. Also in the bacteria, that is the infected bacteria contains the phage material, no, in that only the phosphorus labeled phage materials are released and no this protein coat labeled, that is sulfur labeled structure, that is sulfur labeled phage, that is protein coat is not seen in that bacteria. So only the characteristics of phosphorus labeled radioactive isotope phage material is released from the infected bacteria. So this proves that DNA is the only genetic material, not the protein. So listen students, if they ask this question, you draw this diagram and you explain according to this itself. So you get 5 marks. So I will read it. You will get a clear idea of the explanation with the diagram what I say. All nucleic acids contain phosphorus and proteins contains sulfur, that is in all nucleic acid, that is in DNA you have phosphorus, protein and sulfur in the form of cysteine and methionine. So Hershey and Chase, okay, use the radioactive isotope sulfur S35 and phosphorus 32 to keep separate track of viral protein and nucleic acid during the infection process. Okay, the phages were allowed to infect bacteria in culture medium which containing the radioactive isotopes 35S, that is 35 sulfur or 32 sulfur. The bacteriophage that grow in the presence of 35S had labeled proteins and the bacteriophage grown in the presence of 32 phosphorus had labeled DNA. So the differential labeling thus enabled them to identify DNA and proteins of the phage. So Hershey and Chase mixed the phages, that is unlabeled E. coli, mixed the phages with unlabeled E. coli and allow the phages to attack and infect the genetic material. Soon after infection, the bacterial cell lyses. So the bacterial cell was gently agitated in a blender to loosen the adhering phase. So it was observed that only 32P was associated with the bacterial cell and 32S in the surrounding material, not in the bacterial cell. So when the phage progeny was studied, the radioactive, it was found that it was carried only 32 phosphorus, not 35S. So thus these results clearly indicate that only DNA, not protein coat, entered the bacterial cell. Okay. So Hershey and Chase thus concluded, that is conclusively proved that it was the DNA, not the protein, which carries the hereditary information from virus to bacteria. So it is an important 5-mark question. You draw the diagram and explain according to the diagram. So you no need to read these things and all. If you understand the diagram with what I had explained you can write it on your own. Now the chemistry of the nucleic acid. So as we concluded, the nucleic acids are the DNA. That is, the nucleic acids are the genetic material. So the nucleic acid is made up of RNA or DNA. And it is a very long chain made up of nucleotides. So each nucleotide consists of 3 parts: nitrogen bases, pentose sugar, phosphate group. Okay. So here I had given the structure of a DNA, that is the DNA structure and analysis. Here you can see, as I said, the DNA is made up of so many nucleotides. Okay. So this is a 3H, that is 3H end. So that is phosphorus, or here we see here. So this is a phosphorus, sugar and a nitrogen base. So phosphorus, sugar and nitrogen base forms a nucleotide. Okay. Nucleotide, and the sugar and the nitrogen base forms the nucleoside. Okay. Now the nitrogen base means adenine, thymine and guanine, cytosine in DNA. In RNA, instead of thymine, uracil is present. Okay, in RNA. And remember, the adenine and guanine are purines. And guanine and adenine are purines. And thymine and cytosine are pyrimidines. The adenine always pairs with double hydrogen bonds with thymine. So adenine always pairs with thymine with 2 bonds. Like that, guanine, that is guanine, always pairs with cytosine with triple bonds. So you have seen this condition in last year class in your 11th standard, and you have studied about the, what is that, Chargaff's rule also. So now you must know there are 4 nitrogen bases: adenine, guanine, cytosine, thymine in DNA. In RNA, instead of thymine, uracil was there. That means you have 4 nucleotides. That is, adenine is the nitrogen base, and pentose sugar and phosphorus. So adenosine triphosphate forms 1 nucleotide, and guanine, sugar and phosphorus from guanosine phosphate. So nucleotides, like that it is formed. Okay, 4 different types of nucleotides are formed, and the backbone of the DNA is the phosphate. So now we will see the pentose sugar first. So there are two types of nucleosides, that is RNA and DNA. So deoxyribonucleic acid contains deoxyribosugar and ribonucleic acid RNA contains ribosugar. The only difference between deoxyribosugar and ribosugar is the one, that is you can see a hydrogen with a box in the deoxyribosugar and in the ribosugar. With the elimination of oxygen, in ribosugar you have OH group in the same place, but here the O is missing, that is the oxygen group is missing, one oxygen group is missing. So deoxy means elimination of oxygen. Okay, so because of that only the deoxyribosugar is seen in DNA and ribosugar is seen in RNA. Both are pentose sugar only. Nitrogen bases. As we discussed about, there are 4 nitrogen bases which is classified into two purines and 2 pyrimidines. Adenine, guanine or purines. Okay, and they are double carbon nitrogen ring structure. Double carbon nitrogen ring structure. Okay, thiamine, cytosine, uracil have single ring structure. Remember, they may ask in the 1 mark. So why purines means purines have double bond ring structure, but pyrimidine has single bond, that is single ring structure. So double ring structure, double carbon ring structure. Remember, adenine and guanine are purines. They have double carbon ring structure. Guanine, cytosine, uracil have single carbon structure, pyrimidine. So based on this only the nitrogen bases are divided into 2, purines and pyrimidines. So purine has double carbon structure ring and pyrimidine has single carbon ring structure. Okay, so purines are adenine, guanine and pyrimidines are cytosine, thiamine and uracil. So thiamine is seen only in DNA and uracil is seen only in RNA. The phosphate functional group. So there is a phosphoric acid. Okay, phosphoric acid to form the formation of the strand. As I said in the beginning, phosphate is the backbone of the DNA, that is genetic material DNA and RNA. Okay, it only forms the nucleotide along with the nitrogen bases, okay, and the sugar. So the pH is maintained, that is H plus ion. You have studied the pH in 10th standard and now also you will be studying in your chemistry. So the pH is maintained by the phosphate group PO4 structure. The PO4 structure in DNA and RNA gives the acid, that is nucleic acid, as we say deoxyribonucleic acid. Then acid name was derived from the phosphate group POH by H3 by H plus. So it is named as nucleic acid. Nucleoside and nucleotide. So nucleoside means, that is nucleoside and nucleotide. As nucleoside means sugar and nitrogen base forms the nucleoside. Phosphate group and nucleoside forms the nucleotide. Okay, so in the first line it is here it is nitrogen base. Okay, okay, that is how the nucleotide is formed is the nucleoside, that is the nucleoside is formed. First we will discuss about the nucleoside means nitrogen base plus sugar gives nucleoside. The same way nucleotide, that is nucleotide, how it forms means phosphate group plus nucleoside. Plus nucleoside means nitrogen base and sugar forms nucleotide. And the RNA and DNA are distinct. Why distinct means with the symbol 5-3. So 5- means the carbon sugar which phosphate functional group is attached. So 5' means the carbon group attached to the phosphate functional group. So that means 5'. Okay, 5'. And 3'. 3' means the sugar is attached to the hydroxyl group. Okay, so remember they are distinct with the symbols 3' and 5'. 5' means the sugar is attached with the phosphate group. And 3' means the sugar is attached to the hydroxyl. Hydroxyl means OH group. Okay. That is seen in DNA. In RNA, the OH group at the second position in the ribose sugar. So, in the second position it was attached. So, 5', 3' direction are critical. So, it is very critical and it is useful in replication and transcription, which you going to see in the next class. So, the replication and transcription, it is a critical one, how the direction takes place. For direction only they have given 5', 3', okay. So, based on the X-ray diffraction, that is Wilkins and Franklin's proposed the double helix model, and the replication was given by Watson and Crick, that is they have also given the double helix model, and based on the X-rays diffraction takes place in the bacteria, they had given a replication method also. So, you are going to study in detail the replication and the protein synthesis in the coming classes. So, next you must know, already you have studied this base pair rules or Chargaff's rules in your that is 11th standard. So, you must know adenine always pairs with thymine, that is A double bond T, that is adenine double bond T with 2 hydrogen bonds, and guanine pair with cytosine with triple bonds G triple bond C. So, the ratio between adenine with thymine and guanine with cytosine are constant and equal. So, the ratio is always constant between adenine and thymine and guanine and cytosine. So, it is constant and equal. So, thank you for watching the video. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.